I would like to discuss with you a little bit now what a p-value is and what it is not. I hope I can already calm your worries if I say that I will not provide any formulas in this talk, so don't worry about that. And I would like to start with something I find quite nice. That's a little video that we will watch together here, just 1 minute. It just takes a moment here, sorry. There we go. And here it is. Okay. Enjoy that.
So the question is, what is a p-value?
What's a p-value?
What is a p-value?
What is a p-value?
A p-value.
Oh.
What is a p-value?
I'm going to pass on that.
So, wow, the p-value is
The hypothesis you're testing is
you need statistics to try to estimate if what you think is there.
I know what many people that I have respected have written about and in fact quoted them. Is that a roundabout enough way to dodge your question?
Can you explain what a p-value is in a sentence?
Well, I've actually spent my entire career about the definition of p-values, but I cannot tell you what it means, and almost nobody can.
Okay, I think we all got this. Just a provocative question to everyone. Who of you would have thought they would have had an answer if they had been asked? Just you can use your reactions and just say yes or no, or as we did before. Okay. So far there are more people who would not have dared to answer, but maybe they're catching up. So yeah, the general picture is that more of you would also have passed on that question than would have volunteered in answering it. And basically what we see here, it's scientists who have worked with statistics quite a lot, but they don't manage to give a simple definition in one sentence of what a p-value is. And that is simply because it is not so easy, it's not so simple. And I mean, all of them, they would have been able to give a proper definition like what you read in textbooks or what we will discuss in a minute, but there's no intuitive concept to grasp here. So it's nothing intuitive. So how could we define a p-value? Well, here I will use the ASA statement, so the one Michael was referring to before. And you know, if we want to put it informally, then we could say a p-value is the probability under a specified statistical model that the statistical summary of the data would be equal to or more extreme than its observed value. And so we see here it is a probability, and it exists in the context of statistical hypothesis testing. And what is important is that it's kind of the probability of the data given some model. And that's important. We will get back to that in a minute. If we want to put it even more informally, then we could even shorter say p-values indicate how incompatible the observed data may be with a null hypothesis. This is from the new guidelines for statistical reporting in the New England Journal of Medicine, which I, by the way, recommend very much to you to have a look at that. And so the p-value is a probability, and the value of that probability indicates how incompatible the observed data may be with some null hypothesis. So it's, as this probability exists in the context of statistical hypothesis testing, I would like to just take a look at the principles of statistical hypothesis testing again a bit more in detail than what Michael had outlined before. So how does it work usually, and where does the p-value come into play? So usually, well, hypothesis testing is part of the scientific process. So it's all about learning something from data, and what we want to learn is to kind of generate some scientific knowledge. But of course, we cannot simply use only a few observations to infer some general statement. And therefore, and that's actually, that was something that happened in the field of philosophy of science as well during some time in the beginning of the 20th century, maybe, that something called positivism came in. And the idea was, well, we cannot prove a hypothesis, but we can falsify one. And so that kind of logic of falsifying hypothesis is behind this hypothesis testing. And so the idea was, and that had nothing to do with statistics at that point yet. So it was just that since we cannot prove a hypothesis very easily unless we can observe all objects or subjects it is about, we should try to find hypotheses, formulate kind of null hypotheses, and then reject them. So if we can, it's much easier to reject something. And so in this sense, it works in statistical hypothesis testing as well. So we formulate some hypothesis, some states of the world, and the null hypothesis is actually the one that we want to disprove. And the alternative is the one we're actually interested in. And in clinical science or clinical research, that's often something like, well, there is some kind of treatment effect or a difference between some treatment groups or something like this. That's what we want to show. But to be able to do that, we have to state a hypothesis, the so-called null hypothesis, that there's no effect or no difference, and that's the one that we want to reject somehow. So what I have to explain here still is that in statistics, these hypotheses are not simply the idea that there is a treatment effect or that there is no treatment effect. The hypotheses in statistical hypothesis testing are basically full statistical models. So these are probability models that try to explain how the data that we will gather or will have gathered have been generated. And these include a lot more assumptions that than that there is some difference or not. So that's something to keep in mind. Okay, so if we want to give a little table to show what can happen then in a statistical hypothesis test, we can look at a decision table, because actually we take decisions here. It's a decision problem. We have some hypotheses about the states of the world that should kind of cover the whole range of possibilities. And so the hypothesis can be either true, the null, or the alternative can be true. And the decisions that we can take in terms of generating new knowledge is that we either reject the null hypothesis or we do not reject it. And of course, this is not necessarily correct, the decision that we take. So if we do not reject it and it is actually true, okay, that's fine. But if it was actually true, but we reject it, we have some kind of error. And also, if it is not true, but we do not reject it, we commit another type of error. And these are what I usually call type 1 error and type 2 error. And the type 1 error is also known then as this significance level of a test. So that's what can happen. And so we can think about this in advance. and set these alternatives. And then the idea is that, okay, to decide on this here, I have to collect some data. I want to collect some data to inform my decision. And basically that means that for all hypotheses that you want to investigate, you should actually perform a random experiment, collect some data, and then usually we summarize the data in a suitable statistic. So a statistic is simply some kind of a summary of data. And usually this is a mean value or some kind of a scaled mean in a t-statistic. And then, well, we compute the p-value, I simply write here. But how do we compute it? Well, if we assume this null distribution, so the null hypothesis is a model about how the data have been generated if there was no treatment effect. And so from this, we can derive how a summary of all this data that we collect in a random experiment, how this is distributed in terms of probability distributions. And then we can derive the probability of having observed the value that we actually observed, or a more extreme value under this specific distribution. We could not directly compute that under an alternative, because if our alternative is simply there is an effect, that actually includes infinitely many models, all models where the treatment effect is different from zero. So that's just something to be aware of. And after we computed the p-value, usually we decide to reject the null hypothesis if this p-value is smaller than some pre-specified type 1 error probability we are willing to accept. And well, very often this is just 5%, right? So that's the procedure, how it looks. And I don't want to go into too many technical details here. I would rather like to look at some hypothetical example and show you a bit, give you a feeling of how these quantities behave and why it might not be such a good idea to base all decisions in statistical or scientific knowledge generation on the p-value only. So imagine we are performing some RCT about some surgical procedure, and we want to look at the occurrence of surgical site infections. I'm using this because I've been working on several studies involving this, so it feels like a natural example. And so assume we have 30 patients in each group. And so if we want to formulate some hypothesis, we have to think of, okay, we have a binary outcome. How do we model this in terms of a statistical model? Well, usually for these binary data, there are the binomial probability distributions, and these depend only on the probabilities of the binary event occurring or not. And so if we assume here we have two different probabilities, P1 for, actually it's pi1 for the treatment group and pi2 for the control. And then if we want to formulate that a bit more rigorously as statistical hypothesis, we would say, okay, the null is that the probability of experiencing a surgical site infection is the same for patients in the treatment and control arms. So that means we assume that this difference of pi1 and pi2 is 0. And the alternative would be just a complementary event to that, which means that this is not 0, this difference. And now to get an idea of what happens, I mean, of course, we're not recruiting patients now, but nowadays it's very natural to simulate outcomes of trials to get an idea of how some things behave. So if we assume that pi1 is really 1/6 and pi2 really 3/6, we can simply use a die to simulate the outcomes for 30 treatment and 30 control patients. That would mean we could throw a die 30 times And whenever the die shows a 6 for patients in the treatment arm, we would record that as, oh, an SSI occurred. And whenever it shows any of the other numbers, we would think, okay, no SSI occurred, so that would get a 0 or a no. And for the control arm, it would be whenever we have, for example, 4, 5, 6, then we would count an SSI has occurred. And for the lower numbers, we wouldn't. So we could do that, for example, here with this virtual die. So here we can, we could start with rolling again. So it's a 2 in the treatment arm. There is no event, no event, no event, no event, no event, no event. Ah, there we have an event, and continue like this. I mean, of course, nowadays our computers can do that much faster. And so I will just show you what results we get if we do this a few times and discuss those with you. So now remember, we assume that this probability of an SSI in the treatment arm is only 1/6, whereas it is 1/2 in the control arm. So if we simulate one clinical trial with 30 patients in each group, we get, for example, a point estimate of this difference here of that is here at minus 0.4 approximately. The gray dashed line indicates where the true treatment effect should be. So we would be happy to get a point estimate, which is here the little diamond on the line, on the green line, that this is quite close to this true one, but the null hypothesis would be here the red dashed line. So the null hypothesis would be that there's no difference. So if we were here, we wouldn't know. And so these are the point estimates, and the line is the 95% confidence interval. And here on this, on the left side, I'm also displaying the p-value of a corresponding chi-square test. And so here in that case, we would say, oh, that's great. But, we could also have had this result, still great. Or this, we cross the border, our p-value crosses the threshold. Good again, good again, good again. Oh no, again, no significant result. And if we do that here, I did it 22 times, you can actually see that it varies quite a lot. So we have quite different point estimates for this difference, varying between minus 0.15 to minus 0.5 approximately. And we have several cases where we don't have a significant result. So if we now think of, okay, what would have been if we actually did not have an effect? How would it look under the null? So we could also simulate another experiment where we assume that pi1 equals pi2, so that they are the same and they're both 1/6. What would happen then? Well, now we have that H0, so our null hypothesis value is the same as the true value, because we are now assuming that they are the same. And here in that case, we get a point estimate quite close to 0, and we get a p-value that is 1. So that's mainly because of rounding in R, that it's exactly 1. It would be slightly smaller because the point estimate is not exactly on the null, on 0. And then here, well, that's actually quite far on the side, and it would almost mean that that the control is better, but it's not significant, just not significant. Here we are more closer to what we would like to have or expect. And if we do this 22 times, look at that p-value here. We have our first significant result. And then, again, another significant one. So, of course, that can happen. And so here, what we see here is just results from performing a trial with 30 patients in each group, if they really have the same probabilities for the event. But we still see that actually there's quite a lot of variation. There's a lot of variability. All these 22 here on this slide have been simulated from the same model. But of course, statistical uncertainty means we have some type 1 error probabilities. That's exactly these two here that are significant and that come with that. And that's something very important to understand. So to conclude this part of the example, we've seen that there's a lot of variability, and even though across these 22 in each of the two settings, and that just reflects statistical uncertainty. Of course, here the sample size was rather small with 30, but statistical uncertainty is always involved. We are dealing with probability models, and so everything is true only with a certain probability in our world as statisticians. And you've seen that also the p-value varied quite a lot for, you know, settings that were all simulated from the same true model. The p-values varied a lot. So very often they changed from below to above the threshold, and even if they didn't, they varied quite a lot. So the p-value itself is subject to random variation. Yeah, you might be lucky, you might be unlucky. And the point is, if all conclusions are only based on this p-value itself, on whether you find a statistically significant result, and if you only look at being below or above a certain threshold, this might give you the impression that you find something certain, but you don't, because the p-value itself is is affected by variability. It is an uncertain quantity, so it is not a magic wand that can turn some uncertain results into some certain knowledge. And therefore, we really have to embrace the uncertainty that is underlying all statistical results, all the results of statistical analysis. And this brings us to what a p-value is actually not. And let us have a quick look at what the ASA statement said about that. So it said that it can indicate how incompatible the data are with a specified statistical model. And now it says 5 more points, what it is not. So they do not measure the probability that the studied hypothesis is true. So it is the probability for the data or something more extreme, given the hypothesis, the null hypothesis is true. This cannot simply be turned around and say, well, if I reject the hypothesis, I have some indication of how probable it is. No, the hypotheses are fixed, the probability is about the data. And they clearly say that you should not base conclusions or decisions solely only on whether a p-value passes a specific threshold. You also need full reporting and transparency. Moreover, a p-value does not measure the size of an effect or the importance of a result. Everything will be significant in the end if your sample is large enough, but it doesn't mean it's a big effect. It's an important effect. It's clinically relevant. That is also many important aspects of interpreting results of statistical analysis. And by itself, a p-value does not provide a good measure of evidence. So it just means don't look only at p-values. And to conclude, I have 2 nice quotes for you. The first I like particularly is that the p-value was never intended to be a substitute for scientific reasoning. And that's what Ron Wasserstein said when they announced the ASA statement in 2016. And the second, we have no desire to ban p-values. Instead, we wish them to be considered just as one piece of evidence amongst many, along with prior knowledge, plausibility of mechanism, study design and data quality, real-world costs and benefits, and other factors. Thank you.